So, what is this tissue again? What is the organ? First thing to recognize the organ. Can you somehow send us all the slides? You don't have access to these slides from the Blackboard? I think you do, because they're on the additional materials, which are actually there. Can you try to have some access now, so that I will be sure Blackboard last time we downloaded them? I think they're online. Try to make some access to these. It's on additional materials. You go to microscopic slides, and then they are listed there. You can see them. So, found it. Great. So, take a look at this organ again. What do we recognize here? Not much. A bronchus. Okay. Why? Because of the cartilage here, right? It's centrally positioned. It's purplish. And next to this place, again, one cartilage here, a place which is filled with multiple nests of cells. I'm searching for some portion of normal pulmonary parenchyma to show you here. I only see a typical one. Yeah, there you go. So I see pulmonary parenchyma here, which is a little bit more smashed, suffering complete collapse at some points because it's actually suffering pressure due to this formation of a large tumor field. What we see here are alveolar spaces suffering collapse or atelectasis because they're smashed by the neighbor growing process of tumor formation. And now let's take a look at the tumor here. First thing, what is this field here? Is it recognizable? It's completely amorphous. Some small portions of anthracotic pigment remnant. Acellular. What is this field here? How would you call it? At the center of a tumor nest or something. How do you call it? It's a tumor. Necrosis. No, it's not stroma. Stroma should be something cellular with nuclei. And this is necrosis at the center of a tumor nest. Let's find a smaller nest to explain what is on the inside. That is even more recognizable. Take a look at these. Okay, I will move it slower, sorry. So, what we observe, do you see now well what I'm showing to you? Yeah, it's not, but also it's not as a usual practical. Sorry, is it blurry? Do you see it clear now? Or is it blurry? That's weird. I see it clearly. How are the others? Maybe you should wait for some time up to when it becomes to be observed more clear, or if you want to, open them from the additional materials so that you would be able to observe it better and find the exact field. Now, what we observe here is a nest of cells on multiple layers, which are actually well demarcated. If we observe them closer, we'll take a look at these cells and we'll find that they're all different from each other. They are suffering severe tissue and cellular atypia and nuclear atypia at the mark of Polymorphous cells, hyperchromatic nuclei, dispersed chromatin in certain places. Cytoplasm appears to be more pale at a form of eosinophilic, which means pinkish. Also, the nest is well demarcated, and there are multiple layers of cells which are pretty much resembling the order of squamous epithelium cells. That is how they are getting arranged next to each other on multiple layers. Centrally positioned of this nest, we can observe some other cells which are actually mixed with pinkish sort of substance. This is formation or beginning of a formation of a keratin pearl, which we know that is usual for squamous cell carcinoma. So here, what we observe is formation of nests of cells, beginning of necrosis, keratin pearls, and multiple layers of cells which are situated inside in stroma. So that's the parenchyma of the tumor, which is made again of these atypical squamous cells. And here you should mark all of the signs of cellular and tissue atypia, which you know. What was included when we discussed cellular atypia? What will you mention here? Do you remember it? I think you do. What should we mention about cellular atypia when we discuss a tumor growth? Okay, hyperchromasia, pleomorphism, mitotic figures, dispersed chromatin, nucleoli inside in the nuclei sometimes in some of the tumors, not in all of these. Loss of polarity, well, here not much. They're again situated in multiple layers, but as far as you can see, more the arrangement is haphazard. All of the cells are polymorphic. They're different from each other in size and shape. The nuclei are also different. And if you take a look at the nuclei themselves, they'll be appearing at some points with a very evident nucleola. Here, here, also here. At some slides, it might be even appearing reddish. These are the nucleoli in nuclei with dispersed chromatin in eosinophilic cytoplasm, and they're on multiple layers. What we observe here is beginning of formation of keratin, which again might be observed here. Am I moving too fast? Ah, the share has stopped. The picture is gone. Yeah, I saw this. I will share it again. Sorry. You see it now. Okay. So what we observe here, again, a nest of cells with central inflammatory layer and desquamated cells both, formation of keratin, keratin again. Here, keratin formation of a pearl, nest of cells. Here, hyaline cartilage in the middle of a carcinoma growth. And if we take a look at the slide as a whole, that would be here the normal pulmonary parenchyma, remnants of cartilage. Cartilage is never invaded by carcinomatous growth. Carcinomas grow surrounding the cartilage structures, but they are too hard to be destroyed. Some of the very poorly differentiated structures with multiple necrosis might be able to do it, but most of the times cartilage is spared in between the carcinoma nests. So this again is nest of tumor cells with central keratin production with necrosis. This is a central necrosis in the nest of cells. with hemorrhages here and there. We might be able to observe them. Take a look at these. This is amount of blood which is not positioned in a vessel. So this is a hemorrhage, which is completely normal for types of carcinomas. And here there is a bronchus. That is remnants of respiratory epithelium here. It's filled with fluid, as you can see it. And we might be able to take a look at where the tumor starts exactly. Take a look at the respiratory epithelium and where the squamous cells start to grow, forming this plastic field with centrally positioned keratin, and further developing into squamous cell carcinoma. So we exactly can even observe where the tumor started from. It started from a bronchus, from squamous metaplasia in a bronchus. So we marked the atypical features of the cells. We marked the hemorrhages and the necrosis. Here is a huge field of necrosis. Do not be mistaken to call it keratin. Keratin should be pinkish, and this one is having some sort of shades of cells. Do you see them? This is how necrosis is looking when you observe a tumor. So it's very evident. Also, a hemorrhage here, a remnant of a nest of a tumor, of the tumor actually. Here again, keratin formation. Next to the field of necrosis, take a look at keratin, and necrosis itself. So what you're going to be asked here is going to be actually a question, lots of questions, some of these connected with the idea of how many types of tumors, could start in the lungs. Those would be the adenocarcinomas, the squamous cell carcinoma, the small cell carcinoma, which is actually a neuroendocrine type or non-differentiated type of the other two. What else? Also, different types of mesenchymal tumors, which could also start from the supporting tissues in the lungs. A gross classification is needed for this type of tumors. For your exam, it's given on a slide in your lecture, which you should observe and learn. And what else? Pretty much all, actually. How this tumor could give metastasis, TNM classification, and G at the form of differentiation, also very important. Classification depending on the size of tumor, the engagement of Hiller of lymph nodes, giving close or further metastasis, and differentiation of the tumor. Due to the view, the look of this tumor, what type of differentiation you would be able to give. If you have in mind the information that highly differentiated squamous cell carcinomas are considered to produce keratin and to be shaping or forming nests, and poorly differentiated squamous cell carcinomas never form keratin pearls and sometimes are looking more solid as a position of cells without formation of nests. That would be highly too well differentiated. That's a good answer. Yes, highly too well differentiated type of squamous cell carcinoma. So that's a good answer. Do you have questions for this slide?